Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this particular video, I will be explaining you metabolism of fructose and applied clinical aspects. So I will be discussing how fructose is metabolized into a glycolytic intermediates, and also if there is any deficiency of enzymes that are involved in fructose metabolism, what kind of diseases or metabolic abnormalities that will occur. Let's get into the details. Now the fructose gets into our body through food. So some of the metabolic sources of fructose for us is honey, which contains glucose plus fructose in 50 and 50 ratio in equal concentration, and also high fructose corn syrup, which contains 60% of fructose and 40% of glucose, table sugar, which is basically containing sucrose, and that will be digested in the intestine into glucose and fructose. Now like this, fructose gets into our body and absorbed in the intestine and be transported into the portal system, and from the portal system, most of the fructose is taken up by the liver. Liver is the one that retains majority of the fructose and releases glucose. So that means fructose metabolism will be predominantly going on in the liver, although other organs may participate in fructose metabolism like small intestine and kidney. Now, concentration of sucrose in the blood is lower than the glucose. Now, the concentration of fructose in the blood is lower than glucose, and fructose elicits only modest insulin response unlike glucose. Because when glucose is elevated in the blood, glucose is taken up by the beta cells of pancreas and release insulin in response to that. Similar effect by fructose is less. So that means insulin released from fructose is very less compared to glucose, and that's why fructose is one of the monosaccharide that has been recommended in patients with diabetes mellitus. And also, as I said before, since most of the fructose from portal circulation is taken up by the liver, so liver is the principal organ where fructose metabolism is going on. Now let's get into some of the reactions that are catalyzed to metabolize fructose in the liver. Now here is the metabolic pathway for fructose, a simple metabolic pathway. Only 3 reactions are there, and later the metabolic intermediates of fructose will simply get into a glycolytic pathway. Now, very first enzyme in the conversion of fructose into fructose 1-phosphate, the enzyme is fructokinase. Now this fructokinase, as it is shown here in the reaction, it is going to convert fructose into fructose 1-phosphate. So first carbon of fructose is phosphorylated, where ATP is broken down into ADP, and the phosphate, terminal phosphate of ATP is attached to the first carbon. So this particular enzyme is similar to glucokinase that convert glucose into glucose 6-phosphate. Now fructokinase, it has got high Vmax for fructose. That means as long as fructose is coming into the liver, so most of this fructose is converted into fructose 1-phosphate, and it will be trapped in the hepatocyte or inside the cell. Now, fructose, other than fructokinase, can also be converted into fructose 6-phosphate by hexokinase enzyme. But that reaction is relatively less preferred compared to conversion of fructose into fructose 1-phosphate. Although there is an option for the cell to convert, particularly the peripheral tissues, to convert fructose into fructose 6-phosphate using hexokinase enzyme, because hexokinase has a group specificity. Anyway, so our enzyme of interest here is fructokinase, which is catalyzing a very first reaction in fructose metabolism. Now, once fructose is converted into fructose 1-phosphate, this fructose 1-phosphate will be converted into glyceraldehyde and dihydroxyacetone phosphate. This will be done by enzyme aldolase B. Now, aldolase B enzyme, it is also referred as fructose 1-phosphate aldolase. Make sure you remember the other name of aldolase B enzyme. See, the other name is fructose 1-phosphate aldolase. Now, there are three kinds of aldolase B enzyme that exist in our body. They are aldolase A, aldolase B enzyme, and aldolase C enzyme. Now, aldolase A enzyme is present in most of the tissues. Aldolase B enzyme is present in the liver, skeletal muscle. Aldolase B enzyme is present in the liver, small intestine, and the kidney. Aldolase C enzyme is present in the brain. So, aldolase A, B, and C enzyme, they have got an ability to catalyze fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which is there in glycolysis. But only aldolase B enzyme is the enzyme that can convert fructose 1-phosphate into glyceraldehyde and dihydroxyacetone phosphate. You got to remember this. So that means only aldolase B enzyme has got an ability to participate in fructose metabolism, thereby it can convert fructose 1-phosphate into dihydroxyacetone phosphate and glyceraldehyde. That's why aldolase B enzyme is a rate limiting enzyme in fructose metabolism. That is an important point. It is a rate limiting enzyme because this is the only enzyme that can convert fructose 1-phosphate into glyceraldehyde and dihydroxyacetone phosphate. Now, what happens to dihydroxyacetone phosphate and glyceraldehyde that is made in this particular reaction? Now, the dihydroxyacetone phosphate, it is a glycolytic intermediate, so it enters into glycolysis. Whereas glyceraldehyde, it has to be converted into glyceraldehyde 3-phosphate in order to enter into glycolysis. And that will be done by an enzyme called triose kinase. And the triose kinase, it uses ATP to add phosphate to the third carbon of glyceraldehyde and convert that into glyceraldehyde 3-phosphate. Once we get glyceraldehyde 3-phosphate, so the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, they simply go into glycolytic reactions and continue the glycolysis. So that means fructose, without any regulation, it floods into glycolysis ultimately, because conversion of glucose into glucose 6-phosphate, fructose 6-phosphate, and then fructose 6-phosphate into fructose 1,6-bisphosphate, and that reaction is catalyzed by phosphofructokinase 1 enzyme. It is the most regulated enzyme in glycolysis, and then the glycolytic reactions are going on. So that means glucose is highly regulated, metabolism of glucose. So similarly, metabolism of fructose is not really regulated here, because although aldolase is a rate limiting step, but it is not a regulated enzyme. And that's why whenever we consume a fructose, especially in high concentration, fructose simply floods into glycolysis without being regulated, and dihydroxyacetone phosphate, glyceraldehyde 3-phosphate going into glycolysis makes pyruvate, and excess pyruvate, sometimes it can be diverted into lactate also. And of course, pyruvate going into mitochondrial matrix will be converted into acetyl-CoA, saturated TCA cycle. Acetyl-CoA goes out of mitochondria into the cytoplasm and gets into fatty acid synthesis and cholesterol biosynthesis. So like this, fructose simply floods into the metabolic pathways and create a metabolic abnormalities. Alright, so now some more enzymes that can participate in metabolism of fructose, although three enzymes that we have seen. One is fructokinase, aldolase B enzyme, and then 𝗏𝐻 kinase enzyme, right? So let me just highlight one more time. So the sucrose, which is getting into our intestine through the food, converted into glucose and fructose. Glucose metabolism we know, hexokinase, glucokinase reaction and all that, going into glycolysis, right? Now the fructose is phosphorylated by fructokinase to become fructose 1-phosphate, and the fructose 1-phosphate is converted into glyceraldehyde and dihydroxyacetone phosphate by aldolase B enzyme. Now the glyceraldehyde is converted into glyceraldehyde 3-phosphate by 𝗏𝐻 kinase enzyme. And also note that glyceraldehyde on the other side can also be converted into glycerol by reduction process, that is done by alcohol dehydrogenase enzyme. And glycerol can be converted to glycerol 3-phosphate by glycerol kinase enzyme. And glycerol 3-phosphate can participate in formation of triacylglycerol, because glycerol 3-phosphate provides a backbone for triacylglycerol synthesis. I am going to get into that point a little bit later. Now let me explain you what happens to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate that is made from fructose metabolism. So here is dihydroxyacetone phosphate and the other is glyceraldehyde 3-phosphate. Now the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they both get into glycolysis and ultimately converted into pyruvate. Now what happens to the pyruvate? So pyruvate has got two fate here. Pyruvate can go into mitochondria and form acetyl-CoA, that's one fate of pyruvate. Or pyruvate, whenever this conversion of pyruvate into acetyl-CoA is kind of saturated, at that time when the pyruvate is building up, and also note that when we consume excess sucrose, table sugar, which is increasing fructose, and that excess fructose simply increases pyruvate, because it gets into the metabolic pathway without being regulated. Okay, so that pyruvate, some of the pyruvate is converted into lactate, it is diverted into lactate, and lactate can build up, leading to lactic acidosis. So excess pyruvate can lead to, consumption of excess pyruvate can lead to lactic acidosis. And now pyruvate, which is diverted into acetyl-CoA, acetyl-CoA getting into the TCA cycle, it will combine with oxaloacetate and it makes citrate. And citrate is a TCA cycle intermediate there. So TCA cycle will go on, but TCA cycle can get saturated whenever person is in well-fed condition and have excess NADH+. So when TCA cycle is getting saturated, at that time citrate moves out of mitochondria into the cytoplasm, and in the cytoplasm citrate breaks into acetyl-CoA and oxaloacetate. And this acetyl-CoA now in the cytoplasm is diverted into fatty acid synthesis, leading to increase in fatty acid levels and also cholesterol synthesis. Okay. Now fatty acids are synthesized here in increased concentration and cholesterol is synthesized at high concentration, because fructose just floods into the glycolysis and pyruvate going into acetyl-CoA, saturating TCA cycle and coming out as citrate converted back into acetyl-CoA, and that is diverted into fatty acid and cholesterol formation. And these fatty acids which are formed in the cytoplasm, so they are simply attached with the glycerol 3-phosphate to make triacylglycerol. That's where glycerol 3-phosphate comes in, because I told you conversion of glyceraldehyde into glycerol done by alcohol dehydrogenase, as it is shown here. Okay, let me highlight that. So alcohol dehydrogenase converts glyceraldehyde into glycerol, and glycerol converted into glycerol 3-phosphate by glycerol kinase, especially in the liver. And this glycerol 3-phosphate, it is going to be attached with 3 fatty acids to make a triacylglycerol. And so that means tags will be increased. There will be increase in triacylglycerols in the liver. And these triacylglycerols will be loaded to ApoB100 protein in the liver, and that leads to increase in VLDL formation, very low density lipoprotein formation. And this with these very low density lipoprotein levels will increase in the blood, and very low density lipoproteins they are rich in triacylglycerol, and that's why consumption of high fructose will give rise to hypertriacylglycerolemia, excess glycerol level, triacylglycerol levels in the blood, called as hypertriacylglycerolemia. Okay, in the same way, the cholesterol that is made from acetyl-CoA in the cytoplasm, because I told you, so this acetyl-CoA in the cytoplasm, it is diverted not only into fatty acid synthesis, it is also diverted into cholesterol formation. And this cholesterol which is made in the liver, so it will be loaded onto again ApoB100, and that becomes part of VLDL, and excess VLDL in the circulation, that is also contributing to hypercholesterolemia. Excess cholesterol in the blood is hypercholesterolemia. So basically, consumption of excess table sugar which contains sucrose and giving fructose, excess fructose in the liver especially floods into glycolysis and later into fatty acid synthesis, cholesterol synthesis, making glycerol 3-phosphate available on the other side, so ultimately increasing VLDL levels. And when the VLDL levels are increased in the blood, it is contributing to hypertriacylglycerolemia and hypercholesterolemia. And also on the other side, when pyruvate is kind of diverted, means when pyruvate is building up, it is diverted into lactate form. Edit this. So, and also note that when pyruvate is building up and some of the pyruvate is diverted into lactate formation, lactate builds up in the blood, giving rise to lactic acidosis. Furthermore, what happens here is when fructose is converted into fructose 1-phosphate, and that fructose 1-phosphate in the very first reaction, and the fructose 1-phosphate has to be broken down into glyceraldehyde and dihydroxyacetone phosphate done by aldolase B enzyme. So, when the aldolase B enzyme gets saturated, at that time what happens, some of the fructose 1-phosphate kind of builds up, and also consuming excess fructose is using the ATPs to convert that into fructose 1-phosphate. Remember that. So there is high consumption of ATP there to make fructose 1-phosphate. So there can be a lack of ATP. Eventually it can lead to lack of ATP, right? So shortage of ATP in the liver, and that can also affect metabolism in the liver, and ATPs can be continuously broken down into ADP here, because ATPs are continuously converted into ADP by fructokinase enzyme, right? And that ADP further can be converted into AMP. ADP can be converted into AMP, and AMP can break into adenine, and adenine ultimately going to breakdown process to make uric acid. So there will be increase in the uric acid if there is excess consumption of fructose, because adenosine triphosphate is used here, converted to adenosine diphosphate, and adenosine diphosphate, if it undergoes breakdown process, it leads to uric acid formation, because ultimately purine adenine will be converted to uric acid in the degradation pathway. So hyperuricemia, patient will show, not really patient, even normal individuals, if they consume too much of fructose, it may lead to hyperuricemia. That's why excess consumption of fructose is not a good thing. So this is how the connections can be made here. So what I have done here is I have shown you how lactic acidosis is happening here, how hyperuricemia can be the result of excess fructose, how hypertriacylglycerolemia can happen and how hypercholesterolemia can happen because of consumption of excess fructose. Alright, so that is the metabolism of fructose. So, genetically there can be deficiency of certain enzymes which are involved in fructose metabolism, and two of those genetic deficiencies are shown in this particular figure, and that is, if there is a deficiency of fructokinase enzyme, it will give rise to a disease called essential fructosuria, and if there is a deficiency of aldolase B enzyme, it will give rise to a disease called hereditary fructose intolerance, sometimes referred as fructose poisoning. Now let's get into those two genetic disorders related with fructose metabolism. So the first one is essential fructosuria, and this is because a deficiency of fructokinase enzyme. So what is the fructokinase job here? So fructokinase normally is going to convert fructose into fructose 1-phosphate, and during this process ATP is used and released as ADP. So basically terminal phosphate of ATP is incorporated into the first carbon of fructose to make fructose 1-phosphate. Now the fructose 1-phosphate gets into further metabolic reaction there. Now the deficiency of this fructokinase here will simply build up fructose in the tissue, and later it will appear in the blood. So that leads to fructosemia, means excess fructose in the blood is referred as fructosemia. Excess fructose in the blood, and this fructosemia, because fructose is water soluble monosaccharide, so this fructose can appear in the urine because fructose do not have any renal threshold. So there is no renal threshold for fructose, it simply appears in the urine, and that condition we call it as fructosuria. It is a seemingly benign condition. Patient remains nearly asymptomatic, other than sometimes where elevated fructose in the urine may lead to urinary tract infection. So other than that, generally patients do not show any signs and symptoms. It's an asymptomatic condition. And also the fructose, some of the fructose that is building up in the tissue, it can be metabolized by hexokinase, especially in the peripheral tissue. Hexokinase can convert some of the fructose into fructose 6-phosphate. But this is just a slow phosphorylation process. So still patient will show elevation of fructose in the blood. And also note that hexokinase, it can work only in the non-hepatic tissues because liver do not have hexokinase. It has glucokinase, which do not phosphorylate fructose. And note that liver is the major organ where fructose metabolism is going on. So these are some of the points related with essential fructosuria. Now let's move on to see hereditary fructose intolerance. Now the hereditary fructose intolerance is the most symptomatic condition with fructose metabolism. So here aldolase B enzyme is the one that is deficient. Now what is the job of aldolase B enzyme in fructose metabolism? Let me write down that now. Fructose converting into fructose 1-phosphate, where ATP is used and coming out as ADP there. Fructose 1-phosphate breaks into two molecules, that is glyceraldehyde and dihydroxyacetone phosphate, and that is done by aldolase B enzyme. And also note that the aldolase B enzyme is referred as fructose 1-phosphate aldolase. There are two names here. Alright, now glyceraldehyde can further be converted into glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate, glyceraldehyde 3-phosphate simply goes into glycolysis, as I have explained in the previous slide. Now, what happens if there is a mutation in a gene that is coding for aldolase B enzyme? What if this enzyme is not working? So that will lead to accumulation of fructose 1-phosphate. And this fructose 1-phosphate accumulation, basically it is going to trap this inorganic phosphate, because inorganic phosphate which is there, which is attached to the first carbon, it came from ATP. ATP converted into ADP in the very first reaction, and the phosphate is there in fructose 1-phosphate. Note that phosphate that is getting into metabolic intermediates, it has to be recycled so that inorganic phosphate pool in the cytoplasm can be maintained. So what happens in aldolase B deficiency is simply fructose 1-phosphate keeps on increasing, and that will trap inorganic phosphate. So that's why in hereditary fructose intolerance, there will be decrease in the availability of inorganic phosphate in the cytoplasm, because there will be trapping of inorganic phosphate as fructose 1-phosphate. Because of this, what happens? All the processes that are dependent on inorganic phosphate will go down. So what are all the processes that are dependent on inorganic phosphate directly or indirectly? So glycogen degradation under fasting condition done by glycogen phosphorylase enzyme. You know, phosphorylases, they will add inorganic phosphate to break the molecule, right? So glycogen phosphorylase needs inorganic phosphate to break glycogen under fasting condition and maintain a blood glucose level under fasting condition, which is influenced by glucagon, right? So what happens if there is a deficiency of inorganic phosphate? So glycogenolysis do not go on sufficiently, because glycogen phosphorylase do not get that inorganic phosphate, because most of the inorganic phosphate is trapped as fructose 1-phosphate. So because of this, what will happen? There will be decrease in glycogenolysis. And also, because of lack of inorganic phosphate, even electron transport chain will decrease. Why? Because ATP synthase complex in the electron transport chain, that also needs inorganic phosphate. So the lack of inorganic phosphate will decrease ATP synthesis, and the processes that are dependent on ATP, they will also decrease. One of the process that needs ATP is gluconeogenesis, a new glucose formation under fasting condition. Gluconeogenesis will decrease, and overall, because of this, when glycogenolysis, breakdown of glycogen, when it is decreasing and when gluconeogenesis is decreasing, so overall that will lead to fasting hypoglycemia in these patients. Note that when electron transport chain is decreased, so when there are less ATPs are coming from electron transport chain, so the ADPs that are kind of building up here, because ATP is converted to ADP, so ADP is not converted back into ATP in the electron transport chain. So what happens? So ADP is converted into AMP and releasing that inorganic phosphate there, just to kind of give the inorganic phosphate to a certain extent. Now AMP further, it will break into adenine, adenosine, and ultimately giving rise to adenine, and adenine go into uric acid pathway. So there will be formation of uric acid. Excess uric acid will be formed because more and more ADP is diverted into purine breakdown process, because adenosine diphosphate, adenosine triphosphate. So this is an adenosine, it's a purine. So purine degradation is increased, leading to uric acid. So that patient will show hyperuricemia. There will be hyperuricemia in this condition. Okay, and so patient will show hyperuricemia. And also note that in hereditary fructose intolerance, when dihydroxyacetone phosphate and glycerol D8 phosphate, which is flooding into glycolysis and all that, so overall there can be metabolic abnormalities that is induced here. And the electron transport chain, when it is decreasing, so that will lead to elevation, because NADH plus that has to go into electron transport chain, so that is not going on here. So that leads to increase in NADH levels in the mitochondria, and increase in NADH level in the mitochondria will have a negative effect on TCA cycle, especially on succinate dehydrogenase step, alpha-ketoglutarate dehydrogenase step, and malate dehydrogenase. And also increased NADH will have a negative effect on pyruvate dehydrogenase enzyme, and the pyruvate dehydrogenase enzyme is decreasing its function, and that will lead to elevation of pyruvate, because pyruvate is not sufficiently going into acetyl-CoA, because excess NADH plus having a negative effect on pyruvate dehydrogenase enzyme complex. Why excess NADH? Because electron transport chain has decreased due to lack of availability of inorganic phosphate, leading to elevation of NADH plus, and that NADH plus decreasing pyruvate dehydrogenase complex leads to momentary elevation of pyruvate in the mitochondria. That will get into the cytoplasm, and in the cytoplasm pyruvate is diverted into lactate, and that's why there will be buildup of lactate in the tissue, later appearing in the blood, and that will give rise to lactic acidosis. So patients with hereditary fructose intolerance will show lactic acidosis also. So like this, we can make connections where one abnormality here, that aldolase B enzyme that is deficient, is it just creates a metabolic havoc. It creates a metabolic abnormality, giving us to so many signs and symptoms. Okay, and that is what I have written here. So in hereditary fructose intolerance, patient will show fasting hypoglycemia, hyperuricemia, lactic acidosis. So it is all directly or indirectly because of trapping of inorganic phosphate, making a decrease in cytoplasmic pool of inorganic phosphate. Note that hereditary fructose intolerance patients will show signs and symptoms whenever they consume sucrose or fructose containing food, like consume fruit juice or eat fruit. That's why over a period of time, these patients, they will develop a distaste for fruits and fruit juices. They don't like fruit and fruit juice because every time they consume fruit and fruit juice, which contains basically anything that contains sucrose or fructose, they become symptomatic. Okay, so these are, and eventually because of all these metabolic changes, especially in the liver, patient may show hepatomegaly, patient may show jaundice, and ultimately leading to liver failure and kidney failure. So this is what happens in hereditary fructose intolerance patients. So that's what I have written here. So patients with hereditary fructose intolerance, they will develop distaste for fruits and fruit juice, that's why it is called as fructose intolerance. They are not able to tolerate fructose. Patient will show liver and kidney damage, jaundice, hepatomegaly, cirrhosis of liver, and death usually occurs because of liver and kidney failure. Now, fructose 1,6-biphosphatase, a deficiency of this gluconeogenic enzyme causes fructose intolerance similar to aldolase B enzyme deficiency. Because fructose 1,6-biphosphatase enzyme is a gluconeogenic enzyme which will convert fructose 1,6-bisphosphate into fructose 6-phosphate. So deficiency of fructose 1,6-bisphosphatase, it will lead to simply accumulation of fructose 1,6-bisphosphate. That means two phosphate molecules are attached to this fructose 1,6-bisphosphate. So there are two phosphate molecules that are trapped here as fructose 1,6-bisphosphate, and again the same consequences that I have explained with hereditary fructose intolerance. It's all about trapping of inorganic phosphate, making inorganic phosphate less available for all the processes that needs inorganic phosphate. Okay, so you can use this particular disease as a differential diagnosis for hereditary fructose intolerance, and the name of the disease is fructose 1,6-bisphosphatase enzyme deficiency. Now, what is the metabolic effects of excess fructose? So the excess fructose, simply it bypasses the fructose phosphofructokinase 1 enzyme reaction in glycolysis. It just floods into glycolysis, and more fructose means more and more fructose 1-phosphate, and then going into glycolysis. So simply there is too much of fructose going in, and since fructokinase has a high Vmax, it is converting fructose into fructose 1-phosphate. Okay, and then the fructose 1-phosphate going into dihydroxyacetone phosphate and glycerol D8-3-phosphate done by aldolase B enzyme. So that particular enzyme, it has got a low affinity for fructose 1-phosphate, compared to fructose 1,6-bisphosphate in glycolysis. That means not entirely fructose 1-phosphate is going into fructose metabolism as fast as fructokinase enzyme reaction. So, since it's a slower process, so there can be accumulation of fructose 1-phosphate only when there is excess consumption of fructose, and that fructose 1-phosphate will temporarily lead to decrease in the available inorganic phosphate, and that decrease in the available inorganic phosphate may lead to lactic acidosis, hypertriacylglycerolemia, hyperuricemia, liver damage, kidney damage, hypercholesterolemia, and all those conditions that I have explained with hereditary fructose intolerance. Similar conditions, metabolic conditions can happen even in a normal person consuming excess fructose in the food on daily basis. Now, other use of fructose is formation of amino sugars. Now, as you see here, some of the fructose which is converted into fructose 6-phosphate done by hexokinase enzyme, and also glucose which is converted into glucose 6-phosphate which is converted into fructose 6-phosphate, because in the glycolysis glucose 6-phosphate, which is a glycolytic intermediate, converted into fructose 6-phosphate. So, fructose 6-phosphate can come from glucose as glucose 6-phosphate and fructose 6-phosphate, or fructose can be converted into fructose 6-phosphate by hexokinase enzyme, although it is not really the preferred reaction there. Now, what happens to that fructose 6-phosphate? Fructose 6-phosphate, as it is shown here in the pathway, it is taking a amino group, side chain amino group from glutamine and becoming glucosamine 6-phosphate. Remember this molecule. And the glucosamine 6-phosphate is the donor of all the amino group present in the amino sugar, and ultimately that amino group actually came from glutamine. So, glutamine is the one that gives that is the source of amino group that you see in amino sugars in our body. So, glutamine giving that amino group to fructose 6-phosphate to become glucosamine 6-phosphate. Glucosamine 6-phosphate is acetylated using acetyl-CoA to make N-acetyl glucosamine 6-phosphate, and that N-acetyl glucosamine 6-phosphate will be ultimately going into UDP N-acetyl glucosamine, UDP N-acetyl galactosamine, and they both can go into glycoprotein, proteoglycan, glycolipids, ganglioside formation. So, point here is all the amino sugars that you see in glycoproteins, proteoglycan, glycolipid, sphingolipids, gangliosides, all of them, the source of amino group in them is from glutamine, and it just gets into these pathways in the form of glucosamine 6-phosphate, which is acetylated to become N-acetyl glucosamine 6-phosphate, and ultimately becoming N-acetyl glucosamine, which can be converted into N-acetyl galactosamine. So, have an idea on this just in case if this type of concepts are tested. So, what I have written here is glutamine provides the amino group, glucosamine 6-phosphate forms its acetylated derivatives, and these acetylated derivatives as UDP N-acetyl glucosamine, UDP N-acetyl galactosamine going into glycoprotein, proteoglycan, and glycolipid formation. So, that's about fructose metabolism, all the details, enzymes, important enzymes in fructose metabolism, and also I have explained you what happens if there is a fructokinase enzyme deficiency, aldolase B enzyme deficiency, what are the metabolic consequences of it, and what happens when we consume excess fructose in our body on daily basis. What is the source of amino sugars present in these molecules? It is the amino group is from glutamine, and the molecule that gets into glycoprotein, proteoglycan, glycolipid, ganglioside formation is glucosamine 6-phosphate. That's all about it. I hope this video has helped you in understanding this concept. Thank you.